Look at the next question, student. 16th question. Choose the correct pair from the following. They're given different type of enzymes here and they're asking you to identify the correct pair. Nucleases. How many of you remember in pancreatic juice there is a nuclease enzyme called DNAs, RNAs which digest the nucleic acids, right? Separate two DNA strands. Two DNA strands is separated by the nuclease enzyme? No. Helicase enzyme? Fine. So this is not a correct pair. Exonucleases. Exo means outside. Endo means inside. Right? Exonuclease enzyme make cuts at a specific position within a DNA. Within a DNA means what? Endo. So it is not correct. Right? So this is not correct pair. Ligases. How many of you remember? Ligase is an enzyme which can join the two DNA particle, two DNA molecules. Yes or no? Look at here. Ligase enzymes can join the two DNA molecule. This pair is appropriate. Correct. So look at the fourth one. Polymerase. Polymerase enzyme. Polymerase DNA polymerase 3, which is essential enzyme for the synthesis of new nucleotides during in a DNA replication, right? So DNA polymerase enzyme break the DNA into the fragments. DNA breaking by the enzymes called restriction endonuclease enzyme. So this pair is also not correct. So the correct option of 16th question is answer 3. Ligase enzymes can join the two DNA particles. Okay. So the next question, look at the 17th question. Ray florets are present in sunflower. Right? So option 1, hypogynous ovary. Hypogynous ovary, the ovary occupy the highest position. This is also known as the superior ovary. Example, Malvaceae family. Option 2, half inferior ovary. This is also called what? Perigynous. Perigynous flower. Right? And perigynous flower example we can say the peach and also rose plant. Inferior ovary, you can locate it in, you can observe in guava, apple, cucumber and also sunflower. From the above ovary, right, calyx, corolla, androecium can arise. So ovary occupied, from the above ovary, the organs will arise. So we can say G and dash up on. So this is inferior ovary. Inferior ovary can located in sunflower or guava. We can say best example is apple. So ray floret shows inferior ovary. So the correct answer of 17th question is 3. Inferior ovary. Look at the next question, students. 18th question. Match the organism with its use in biotechnology. So they're given the different type of organism in column 1 and they're asking its use in biotechnology, right? First, bacteria. Bacillus thuringiensis. How many of you remember Bt bacteria live in soil? Bt bacteria has cry protein synthesis gene, right? So that cry gene undergo transcription and translation to produce a protein called cry protein that is very, very sensitive to what? Insects. Right? So I'm going this Bacillus thuringiensis, transgenic plants like Bt cotton and Bt brinjal, Bt tobacco also, right? So Bacillus thuringiensis I'm going with cry protein. So option here 4. Option B, Thermus aquaticus. Do you remember this bacteria? Thermus aquaticus is a one type of archaebacteria which can live at 100 degrees Celsius of temperature. From this bacteria we can extract Taq polymerase enzyme, right? So that Taq polymerase enzyme can be used in PCR method to synthesis new nucleotides. So Thermus aquaticus, I am going with what? DNA polymerase. What type of DNA polymerase here? We can clearly write the Taq polymerase. Fine. So look at the question option 3. Agrobacterium tumefacium. Agrobacterium tumefacium bacteria is also called the TI plasmid bacteria. Tumor-inducing plasmid. In biotechnology, Agrobacterium tumefacium bacteria is very essential to insert our desirable gene in plant cell. So we are using this bacteria as a vector, right? TI plasmid it is also called. tumor inducing plasmid. So option C, I'm going with cloning vector. Here it is 1. So look at the D, Salmonella typhimurium. In the year of 1972, there was 2 scientists called Cohen and Boyer. For the first time, they constructed rDNA, right? So Salmonella typhimurium, they constructed rDNA. So first constructed rDNA, I'm going with Salmonella typhimurium. So here I can write the 2. So final answer is 4, 3, 1 and 2. Where is the 4, 3, 1 and 2? 4, 3, 1 and 2. So the question number 18 answer is 4. So 4 is the right answer for 18th question. Look at the next question, 19th question. The products of reaction catalyzed by nitrogenase in root nodules of leguminous plants. This question from a chapter called Mineral Nutrition, right? They're asking in leguminous family, root nodule, nitrogen fixation. So in this question, we need to understand something. They are already given that nitrogenase enzyme. Nitrogenase enzyme cannot activate it in presence of oxygen. So remove one option here. What's the option? First option, ammonia and oxygen. So it is not a correct answer because nitrogenase enzyme only activated in absence of what? Oxygen. Leg hemoglobin is a red color pigment present in the roots of what? Leguminaceae family. So that leg hemoglobin act as an oxygen scavenger and they create anaerobic condition in the root nodules, right? In the root nodules, right? So look at here. Ammonia and oxygen, so remove that one. It is not a correct answer. Actually, in this question, what they are asking? Reaction catalyzed nitrogenase in a root nodules, which means they are asking the nitrogen fixation, the conversion of atmospheric nitrogen into ammonia. They are asking ammonia, right? This is the nitrogen fixation they are asking. Conversion of atmospheric nitrogen into ammonia. So which enzyme is that one? Nitrogenase enzyme. How many of you remember? In presence of nitrogenase enzyme, look at here. This is the nitrogenase enzyme. We can say we represent like this, right? This is the nitrogenase enzyme. So first what happened? Atmospheric nitrogen with how many bonds? 3 bonds between the 2 nitrogen, right? So this is the nitrogenase enzyme. So atmospheric nitrogen molecule binds to the specific position of a nitrogenase enzyme. Next, here two hydrogen atoms are going to add to the nitrogen, right? So what happened here in the next condition? This is the nitrogenase enzyme, right? So one bond will break between the 2 nitrogen atom. So one pair of electron free here. So one hydrogen atom can bind with each nitrogen, right? So look at here. Here we can say this is one hydrogen here and this is another hydrogen. Again 2 hydrogen molecule will help. Add here, it is also reduction here, right? Reaction, 2 H plus ions, right? One more bond will break there. Look at here. If you take the next reaction here, this is nitrogenase enzyme. One more bond will break here. So 2 bonds. First initially 3 bonds and next 2 bonds. Next 1 bond. So here it is 1 and 1 more hydrogen. 2 hydrogen will add to the 1 nitrogen. Here also 2 hydrogen to each nitrogen. Eventually, come on, tell me here, to the nitrogenase enzyme, 3 hydrogen attached. So from the atmospheric nitrogen, eventually it converted into ammonia. 2 molecule of ammonia in presence of the enzyme called what? Nitrogenase enzyme. Finally, nitrogenase enzyme can set free. Right? So Among these 4 options, which option is the correct? Ammonia and oxygen. In this one, in this entire process is in absence of oxygen, not in presence of oxygen. It is in absence of oxygen. So this is not correct answer. Next, ammonia and hydrogen. Look at here. Ammonia and hydrogen ions are participating, right? Next, ammonia alone, nitrate alone. So the correct answer of this question is what? Ammonia and hydrogen. So the correct answer we can say it is, answer is 2. 2 is the right answer for question number 19. Next question, question number 20. Name the plant growth regulator. They're asking about the plant hormones. Name the plant growth regulator which upon spraying, look at here, they're spraying on which crop? Sugarcane crop. Increase the length of stem. Increase the length of stem and increase the yielding of what? Sugarcane crop. So question option 1, ethylene. Ethylenes are used for the ripening of fruits. It is a gaseous form of plant growth hormone, right? So ethylenes are mainly useful what? Useful for ripening of fruit. So this is not correct. They're asking in the question, increasing the length of stem or elongation of an internode, which is the function of what? Gibberellic acid, right? So look at here, abscisic acid, growth inhibitory hormone and promote seed dormancy. So this is not increase the height of the stem. Number 3, cytokinins, cell divisional hormone. Last one is gibberellins. Gibberellins are also participate in, so when we spray the gibberellic acid in the sugarcane crop, the height of the length of the stem will increase and it will give a good harvest. So the correct answer of this question is what? Gibberellins. Question number 20, answer is 4. Look at the question number 21. Next question 21. The body of the ovule is fused within the funicle at. They're asking about the structure of ovule. Look at here. Let me draw this structure. So this is a placenta where the ovule is going to attach and it has a funicle. This structure is called anatropous ovule I am drawing. So look at here. This is the body, right? So this is the body. This structure is known as funicle, right? It is known as funicle. So they're asking what here? Funicle attached, fuse with the what? The body of the ovule. This is the body. This is known as the body. So body of the ovule attached to funicle at. So keep in your mind, students, here there is the part where the body is going to be attached to the funicle is known as, this part is called hilum. Hilum, right? Next, this is the angiosperm ovule here. So it's surrounded by the 2 integument. It is the bi-integumented, right? So look at here, bi-integums, right? And wherever the integums are not present, this opening is known as micropyle. Micropyle. So inside this ovule, embryo sac is present, right? So this is the embryo sac here. This is embryo sac. These are the cells in embryo sac. Antipodal, synergids, polar nucleus and female gamete. Next, here the diploidal tissue inside the body of what? This ovule is known as nucellus. This is known as what? Nucellus. So nucellus is given. This part is called the chalaza, right? This is called chalaza. Look at here. The body of the ovule fused with the funicle at a specific point known as the hilum. So the correct answer of this question is what? Hilum. Not chalaza, not nucleus, it's not micropyle. So the correct answer of this question is hilum. Question number 21, answer is 3, hilum. So look at the question number 22. The process of growth is maximum during, they are asking. If you remember the geometric growth, it is a sigmoidal curve, right? So look at here, sigmoidal curve. So graph So this is a time. Here it is the height of a plant, right? Height of a plant. So look at here. First phase is what here? This is initially it is a very small. The growth is very small. Initially it is a small. Later it's increase, right? So growth will be increase and at a certain point it will be stable. It will be stable. So the first point where the growth is initial, low growth rate, it is known as what here it is? Lag phase. This is called lag phase. Where the growth is going to be increased, this is where the growth rate is high. This place, this curve is known as log phase. Log phase is also known as what? Exponential phase. And finally, it is a steady phase. Finally, it is a steady phase. What they are asking? The process of growth is maximum. Senescence means death. Senescence means it is a death. So there is no maximum growth. Dormancy. Dormancy means it is a completely inactive of what? All the metabolical process. Right? So there is no growth in a dormancy also. Log phase is the exponential phase where the growth is maximum in a geometrical, what they call growth. So the correct answer of this question is, answer is 3 is the right answer. Log phase is also known as exponential phase where the growth is maximum, right? So the question number 22 answer is 3. Question number 23. Look at the question number 23 from the chapter called Animal Kingdom from 11th grade. Bilateral symmetrical and acyclomate animals. Wait. Bilateral symmetrical animals. Look at what they're asking. Number 1, bilateral symmetrical and acyclomate animals. Coelom is absent. Number 1, Aspirants. In Aspirants, pseudocoelom is present. They are bilateral. Okay, I will agree with this one, but they are bilateral, but they are not acyclomate. So this is not correct answer. This is not correct answer. Annelida, bilateral symmetrical, fine, but they are not acyclomate, they are schizocoelom. True coelom is present in Annelida. So this answer is also not correct. Ctenophora are not bilateral symmetrical, they are what? They are radial symmetrical animals. And acyclomate, okay, I'll agree with the acyclomate, but bilateral symmetry is not correct for what? Ctenophora. So this is also not correct. Platyhelminthes organism, flatworms in an animal kingdom. First time it has been formed, third germ layer, diploblastic, and also acyclomate. Coelom is absent in platyhelminthes. So the correct answer of this question is 4 is the right answer. Bilateral symmetrical and acyclomate is in platyhelminthes. So the correct answer of 23 is 4. Look at the 24th question. 24. Microbes in human welfare. Which of the following is put into anaerobic sludge? Anaerobic sludge. Digester for the sewage treatment they are asking. Primary sewage, secondary sewage. In primary sewage, physical removal of what? Particles. Removal of physical particles and also sedimentation. 2 steps are there. One is filtration and one is sedimentation, right? In primary treatment. Fine. So if you take, this is after filtration, this is a complete water here. If we are keeping this water for some time, the solid particles will come down, right? So these are the solid particles will come, settle down at the bottom of the tank, right? So this solid is known as primary sludge. This is known as what? This is called primary sludge, right? And this, we need to supply this primary sludge into aeration tank. After that, what will happen? Activated sludge. Small amount of activated sludge is again given into the aerobic bacteria. major portion is sending to anaerobic condition, anaerobic sludge. So this is a complete effluent, right? This is completely effluent. So effluent is going into the secondary treatment. So into this effluent, we are giving initially huge amount of aerobic bacteria, and after that it's formed what? Activated sludge. Activated sludge is going to give into what they call anaerobic bacteria. So in which of the following is put into the anaerobic sludge here? Effluent of primary treatment. Effluent of primary treatment is directly given to the aerobic first. Number 2, activated sludge. Yes, after BOD reduce, activated sludge is put into the anaerobic sludge. So the correct answer of this question is what? Activated sludge. Right? So primary sludge, this is a primary sludge, the bottom after the settling of the solid particles and floating bubbles. So the correct answer of this question is the 24th question answer is 2, activated sludge. Look at the question number 25. Question number 25. Match the following columns and select the correct option. Column 1 and column 2. In column 1, option A, floating ribs. In human thoracic cavity, in thoracic place, we have a 12 pairs of ribs are present, right? First 7 pairs of ribs are called true ribs because first 7 pairs of ribs are dorsally they attach to the thoracic vertebra and ventrally they attach to the sternum. So 1 to 7 pairs of ribs are called bicephalic ribs and true ribs. And the rest of ribs are called 8th, 9th, 10, 11, 12 ribs are called false ribs. Moreover, in this question, option A, they are asking floating ribs. So, which ribs are called floating ribs? 11th and 12th ribs are called floating ribs because they are not connected to either sternum or anterior ribs. So, floating ribs, I am going with the 4th option. So, do not connect with sternum. Floating ribs do not connect with sternum. 11th and 12th pair. So I am writing here 11th pair and 12th pair. Option B, acromion process. Where do you observe the acromion process? In scapula flatted bone. If you look at the scapula bone, look at here. This is the triangular scapula bone which is located at the dorsal side of body, right? So this is a flatted triangular bone. And in this bone here, it has a flatted structure and there will be a small projection we known as, and this projection is called the acromion projection, acromion process, where the clavicles attach. So acromion clavicle. I'm going with what? Acromion clavicle. So the clavicle collarbones are auriculated in acromion process of scapula. So if you look at here, this is the scapula bone, right? Scapula bone. So this process is known as acromion process, where the clavicles auriculate. Number C, scapula. Scapula is a triangular flatted bone, dorsally they are present, and if you look at it, the position, the location position is between 2nd and 7th thoracic rib. So I am going with what? Scapula located between the 2nd and 7th true ribs, right? So scapula, I am going with the 1, right? Next, glenoid process. Below the acromion process, there is a small depression called glenoid process. This is known as glenoid cavity. So, in this glenoid cavity, humerus head part will be auriculate. So, the glenoid cavity, into the glenoid cavity, head part of humerus long bone will be auriculated. So, D option, glenoid cavity, I am going with 2 options. So total answer is floating ribs 4, acromion process 3, 1 and 2. So where is 4, 3, 1 and 2? So the correct option of this question is answer 2. 2 is the right answer for the question number 25. Look at the next question, 26th question. Identify the wrong statement with regard to restriction endonuclease enzyme. Restriction enzymes. Restriction enzymes are also known as molecular seizures in biotechnology. Yes. So look at there what they're asking. Wrong statement regarding restriction enzyme. So which statement is not about the restriction enzyme? We need to identify from the options. Option number 1. They are useful in genetic engineering, right? Genetic engineering, the first tool, first major enzyme which cleave the DNA at the specific position is known as what? Specific site is known as restriction enzyme. So restriction enzymes are useful in biotechnology and also genetic engineering. Fine. It's the right one. Sticky ends can be joined by the DNA ligase. Are they asking about the DNA ligase or endonuclease enzyme? Is this the statement is regarding to the endonuclease enzyme? No. So this is not correct. Third option. Each restriction enzyme function by the inspecting the length of DNA sequence. Yes. Fourth option. They cut strands of DNA at palindromic site. This is also absolutely correct. Wrong statement regarding restriction enzyme. So DNA ligase enzyme is not at all related to restriction endonuclease enzyme. So the correct answer of this question is option 2. So this is the right answer. Look at the question number 27. What they're asking in question number 27. Match the following columns and select the correct option. Column 1 and column 2. Look at here. They are asking from the animal kingdom, 11th grade chapter. Gregarious polyphagous pest. This is normally known as Locusta, Arthropoda, Insecta. So this is 4. Option B. Adult with radial symmetry and larva with bilateral symmetry. How many of you remember? In bilateral symmetrical animal, Echinodermata, adult one shows radial symmetry, but the larva shows what? Bilateral symmetry. Yes or no? So keep in your mind. So adult with radial symmetry and larva with the bilateral symmetry is asterisk, sister, Echinodermata. So we can write here 1. Right? Book lungs, Arachnida. Example of Arachnida, scorpion. And also spiders. So book lungs are the respiratory organs in scorpion, Arachnida, Arthropoda. Bioluminescence, emit light in ocean. They are ctenophorans, right? So bioluminescence is the unique characteristic feature in the ctenophora. So what is the answer here? Ctenoplana. Example. So answer is here 3. So 4, 1, 2, 3 is the right answer. So where is the 4, 1, 2, 3? 4, 1, 2 and 3. So the correct answer of 27th question is 4 is the correct answer. Look at the next question, 28th question. If the head of cockroach is removed, it may live for few days because if you look at the central nervous system of cockroach, it has 2 portions. First one called nerve ring. The first part is known as nerve ring, which is surround around the esophagus, and the second one is known as ganglionated ventral tubular nerve cord. Ganglionated ventral tubular nerve cord. So these 2 parts are present in the central nervous system. If you look at the nerve ring, this is the nerve ring which surround around the esophagus of cockroach. It has a principal organ called brain. So in the nerve ring, the principal organ is what? Brain. Students, keep in your mind, in cockroach, brain principally involved in sensory action and also endocrine. Brain has 3 parts, 3 lobes in cockroach. First lobe is known as protocerebrum. protocerebrum. So protocerebrum receive sensory impulse from the compound eye. So they receive sensory impulse from compound eye, right? Now, second lobe is known as deuterocerebrum, which receives sensory impulse from antenna, right? Antenna. Next, the last lobe is known as tritocerebrum. The last lobe is called what? Tritocerebrum. So this tritocerebrum receive sensory impulse from labrum, upper lip. Labrum. So principally the brain is involving in the sensory impulse absorption. Here, brain is present above the esophagus. So, if you remove the cockroach head, it will live a few days because the head holds a small portion of nervous system. In the head part of a cockroach, it holds a small part of nervous system. Rest is situated along the ventral part. Rest is the ganglionated nerve cord. Ventral tubular nerve cord is present in the ventral side of entire abdomen. So if you remove the cockroach will be live for a few days because in the head part, it has a small portion of a nervous system. So it is looking like an appropriate answer. Look at the B. The head holds 1/3 of nervous system, okay? While the rest is situated along the dorsal part. What did I explain? It is not in the dorsal part, it is in the ventral part. So, this option is not correct. Next, the supraesophageal ganglia, which is also known as the cerebral ganglia of the cockroach, are situated ventral part of the abdomen. Students, keep in your mind, cerebral ganglia located below the esophagus. So, it is not present in the ventral part of abdomen. So, statement is correct. This statement is wrong. Fourth one, the cockroach does not have a nervous system. Cockroach have the nervous system. In the nervous system of cockroach, central nervous system, peripheral nervous system, autonomous nervous systems are present. So, what is the correct answer of the question number 28? Question number 28 correct answer is the head holds a small portion of nervous system while the rest situated along the ventral part of the body. That is the reason why if you remove the cockroach head, it will live for few days. So the correct answer of 28th question is option number 1. Look at the question number 29. Which of the following regions of globe exhibit highest species diversity? Simple question. Highest species diversity in Himalaya, Amazon forest, Western Ghats of India and Madagascar. So here in this 4 option, the richest species diversity located in where? Amazon forest. Right? A simple answer, Amazon forest. So the correct answer of the question is 2. 29 answer is 2. Look at the 38th question. Which is the important site of formation of glycoproteins and glycolipid? Look at student here. This is the chapter, this question from the chapter called Cell is the Functional Unit of Life. Formation of glycoprotein and glycolipid in eukaryotic cell. Number 1, Golgi bodies, which we also known as Dalton bodies. Yes? Dictyosomes in plant cell. Golgi bodies are also called dictyosomes in plant cell. So, Golgi bodies in eukaryotic cell play an important role in the synthesis of what? Glycoprotein and glycolipid formation and packaging also, right? Acrosome formation in sperm. Golgi body function. So, Golgi body is the right answer. Polysomes, protein synthesis, endoplasmic reticulum, 2 type of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum, right? Rough endoplasmic reticulum, major function is in protein synthesis, participate in protein synthesis, smooth endoplasmic reticulum synthesis, steroidal hormones such as estrogen and progesterone. Peroxisomes are participate in photorespiration. Do you remember? Peroxisomes are single membrane bound organelles in plant and animal cell. In plant cell, the peroxisomes perform photorespiration. C2 circle. So here they are asking glycoprotein glycolipid synthesis by Golgi body in eukaryotic cell. So the correct answer of the question number 30 is 1. 1 is the correct answer.
Brilliant Qatar is the passport to your success.